Hello everyone, I am Judith Costantine from Dar es Salaam, Tanzania. Welcome to my YouTube channel on pharmacognosy-related matters. So I'll take you through application to biotechnology in pharmacognosy, specifically on plant tissue culture, and this is part 1. So I'll take you through introduction to biotechnology, just a snapshot of it, but basically we are going to touch plant tissue culture in general, see the advantages and disadvantages of plant tissue culture, see the basic requirements for plant tissue culture, the types of plant tissue culture, as well as the basic terminologies that come with plant tissue culture. So you are welcome. Biotechnology is a technology that utilizes biological systems, living organisms, or their parts to develop or make useful products. Please pay attention to these biological systems and living organisms. It makes use of these living parts to develop useful products based on what is needed by that time. So it is a multidisciplinary field. It covers along several disciplines. And one of the key techniques used in biotechnology is genetic engineering. But we are not going to do that. We'll be very specific to tissue culture. So other important techniques used in biotechnology include tissue culture, which allows researchers to grow cells and tissues in the lab for research and medicinal purposes. So plant tissue culture systems represents a renewable source of valuable medicinal compounds. It is a renewable source. We are going to see under the advantages why we do call it a renewable source of these variable medicinal components. So plant biotechnology is the use of tissue culture and genetic engineering techniques to produce plants and that exhibit new or improved desirable characteristics. This is actually what we want in biotechnology. We are interested in desirable characteristics. So it is the generation of useful products or services from plant cells, plant tissues, and often organs. These are very, very small explants we are going to see under the requirements for plant tissue culture. So generally, as I said earlier, we will do much of the plant tissue culture, not everything, but at least to have a light on what happens around plant tissue culture. So generally, plant tissue culture starts with a mother plant. We shall see what a mother plant is, which can be cut into pieces known as explants, and these explants may be treated and introduced in a nutrient medium to get the colors. Then the colors can be, we can play around again with the nutrient media, especially with the growth regulators like cytokinins and auxins to get the plantlets. Then these plantlets may form shoots, then form roots, and then turn into a general plantlet, which is ready for planting. We shall see these steps as we move, but this is just a general picture around plant tissue culture. So plant tissue culture is an in vitro aseptic cultivation of disease-free plant cells, tissues, or organs, and a nutrient culture medium of known composition and environmental conditions for the production of primary and secondary metabolites or to regenerate plant part. So, for example, here, when we talk of plant tissue culture, remember you cannot escape this. It is an in vitro. First, it is done outside the living organs. It is an aseptic technique, and it produces disease-free plants. And it should use nutrient media which has a known composition, and the environmental conditions should be controlled. And in the end, we expect to get either primary or a secondary metabolites or generate a plantlet. So that is what plant tissue culture means. It is generally done in the laboratory to regenerate new plants from mother plants. So it involves the use of small pieces of plant tissues, also known as explants, which are cultured in a nutrient medium under sterile conditions. Like I said earlier, we should do everything under aseptic condition. So what is a mother plant? A mother plant is a plant grown for the purpose of taking cuttings or offsets to grow more quantity of the same plant. So it is a donor of the plant tissues. This is where we get an explant from. And an explant is a piece of meristematic tissue that is used to start the process of tissue culture technique. So a superior mother plant is identified, checked, and verified to have all the growth characteristics particular to the variety. A good mother plant for tissue culture should be healthy, free from diseases and microorganisms, and should exhibit high vigor with juvenile, actively dividing cells like shoe tips, preferred for better, faster regeneration. So ideally, the plant is true to type, genetically stable, and possesses desirable phenotypic traits such as high yield or superior quality. So in order to ensure this aseptic technique, a laboratory disease testing program is recommended to verify freedom from bacteria, from virus, and other disease organisms that are likely to interfere with the process. And what is an explant? An explant is a piece of plant tissue that is placed on the growth medium to initiate in vitro cultures. So this is what we start with, an explant in plant tissue culture. It comes from the mother plant. The very, very required characteristic of an explant, it should be meristematic. It should be able to divide, should be able to differentiate and undergo cell division to produce other plantlets. So it is any meristematic tissue that is extracted from the mother plant and serves as the starting material to initiate in vitro cultures. All parts of plants can be used as explants for tissue culture purposes, provided they are meristematic. That means they should be capable of differentiating and resuming cell division, have cytoplasm. We are going to see what differentiation means. But generally, basically, differentiation means the ability of a tissue to go back, like it grows backward, it turns into a baby so that it may be differentiated to another plant part. So we are going to see what we mean by turning into a baby. So parts of plants like flowers, buds, young embryos, and parts of young seedlings are mostly used as explants for plant tissue culture. So these explants develop into callus, which can be used to regenerate a whole plant. A callus is a group of undifferentiated cells derived from competent source tissues that is cultured under in vitro conditions. When we say undifferentiated cells, this means these cells have not been differentiated into any organ or any part of a plant. It has not turned into roots, it has not turned into shoots, it has not turned into a plantlet. It is just a mass or a group of undifferentiated cells that are ready to turn into anything, depending on how you play around with the composition of the nutrient medium. We shall see all this, don't bother yourself. It's just that you have to make sure you go with me together as we move from part 1 to the last part of plant tissue culture. So some parts like the root tip are rarely used as explants for tissue culture process. Why? Because they have high contamination risk, they are difficult to sterilize, and they have challenging isolation procedures, so we don't prefer using them. And also we don't prefer using flowers because they have this they are rigid to differentiation. They are not ready for that. Most of them cannot differentiate. So what is a callus? We said earlier it's a mass of undifferentiated cells. As you can see there, a callus can be utilized direct to regenerate plantlets or to extract or manipulate some primary and secondary metabolites. So depending on what you want in your plant tissue culture process, you can decide to end up with a callus so that you produce or you manipulate primary and secondary metabolites, or you can decide to play with the composition, especially with the growth regulators, to turn into shoots, roots, and end up with plantlets for further processes. So it depends on what you want. What are the advantages of plant tissue culture? Generally, plant tissue culture enable the rapid year-round production of large number of genetically identical disease-free plants. Yes, we can get plantlets at any time because it is done in the laboratory. Another thing is that it is crucial for conservation of endangered species. Yes, you can agree with me on that, because if we have a sustainable source of plantlets, then we are sure of conserving endangered species. It ensures the availability and the uniformity of raw materials. You can, yes, it has a regular and uniform supply of raw materials throughout. It does not depend on the season, it is not affected by environmental factors and any other thing that is likely to affect the production. It overcomes the fluctuation in supplies and quality, yes, especially on quality. It produces disease-free plants, and plants are healthy. They can grow as much as possible. So that is it. Also, new isolation methods can be accomplished by using plant tissue culture process. Thanks to that, it can be used in tracing biosynthetic pathways for various plants by using labeled precursors in the culture media. It requires small space to grow plantlets by tissue culture techniques. That's it. But everything that has advantages, it also has disadvantages. It ensures production of disease-free plantlets. I've said this before. It enables propagation of plants without seeds, like seedless propagation, and a lot of things. Like I've said this, it is independent on the soil conditions and changes in the climate conditions. I've said earlier. What are the disadvantages of this? It is highly technical, labor-intensive, and very expensive. It is a very expensive process. The key disadvantage include high risk of contamination. So we have to be sure, because this is an aseptic technique, we have to be very, very careful with the contamination. So there is a very, very high risk of contamination, low genetic diversity, potential for genetic mutations, and challenges in adapting fragile plants to natural environment. It is very challenging when it comes to acclimatization of plants produced in the laboratory, to acclimatize them to the external environment. We are going to see what we do with the acclimatization, so don't have to worry about that. A high level of expertise is required. Yes, we have said that, and that goes along with the expensive part of it. A minor error may lead into the complete collapse of the whole process. For example, a minor error in the sterilization process, everything will affect the whole batch. It may need to be recalled. Or yes, it is an expensive process. We have said this earlier in terms of expertise, consumables, chemicals required, and everything around it. There is a chance that the propagated plants will be less resilient to diseases. Yes, they are disease-free, but they are not disease-resistant. So there is a high chance of being less resilient to diseases. And there is no chance for mutation, unlike the seed propagation. In this, there is no chance for mutation, and the protocols for individual plants vary greatly. So it is very hard to have this. Every plant species requires its own protocol. So that brings the expensive part of it for large production of various species of plants. Here are the basic requirements for plant tissue culture. We are going to see this in the next in the coming part. So please make sure you subscribe to this channel and follow along as we move, so that you don't miss these other parts of plant tissue culture. Thank you for now. Please share, subscribe, and like. Make sure you drop a comment, you give us a reference material and anything that we need, and see you in part 2 of plant tissue culture. Thank you for listening. Thank you.